Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Kami Biology. Today in this video lecture, we are going to talk about initiation of DNA replication in eukaryotes. In the previous video lecture, if you guys remember, we talked about the DNA replication introduction in eukaryotes, and we talked about the proteins which are involved in DNA replication in eukaryotes. Okay, so before going to explain initiation of DNA replication in eukaryotes, you guys need to understand that what kind of the protein we needed in DNA replication in eukaryotes. So I already explained it. You can watch it into my channel, or don't worry, I will give the link in the description box, then you will check it. Okay, so let's start the DNA initiation of DNA replication in a detail. So let me remove and let me start. So now let me write initiation of DNA replication in eukaryotes. Okay. Now, how the initiation of DNA replication is started in eukaryotes? First of all, what is initiation of DNA replication? Initiation of DNA replication is the stage where the DNA can decide the replication process, and the initiation is that stage where the DNA replication can be started. Okay, first thing. The second thing is that this initiation stage is very important, not only in replication, it is also important in transcription and translation as well. Why? Because at that initiation the process is started, and due to this stage the next stages is also going on. But once the initiation stage is not start, so remember, without this initiation the rest of the stages not happen, means elongation and termination, which is present also in transcription and translation as well. We will explain it in our future video lecture. Okay, so it is very important stage. And remember, the initiation can be started, means the initiation of DNA replication in eukaryotes can be started at a specific region of the DNA molecule. We call that origin of replication, or we also denoted it with ORIC site. So if we have a DNA, in this case, again let me draw, if let's suppose this is a DNA molecule, now it's one strand going on 5' to 3', while the other is 3' to 5'. Now that specific region is called as origin of replication, or ORIC site. And remember, that specific region is rich with adenine and thymine, means adenine and thymine bases. So that's why we call that AT rich region. Now this DNA is inactive form. So we have a protein that is activated during initiation stage of DNA replication in eukaryotes. We call that protein ORC protein. So first of all, we have a protein that is activated, we call that O or C protein. And we explain that ORC stand for origin recognition complex. It is not only one protein, it is the complex protein. I mean, it contains many of the proteins, ORC 1, 2, 3 and 4, and it can make ORC, means origin of recognition complex. That's why we can say that it is a complex, mean it is the combination of different protein. So ORC is activated and bind at that specific site. So let me draw here again the DNA molecule, just a little bit large. Okay, so this is not the replication bubble. Okay, or then this diagram doesn't mean that DNA can meet. Now I just draw here a little bit larger. And one thing more, remember that origin of replication, we have not only one origin of replication in the eukaryotic. We have one origin of replication in case of the prokaryote, but in case of the eukaryotes we have different and many of the origin of replication. But here I just draw one, just for the explaining the process of the initiation. Okay, now the ORC is activated and it can bind at that specific site, ORC. and that specific site is the origin of replication. Okay, now when the ORC can bind here, then after that they can activate it to other proteins, and they can give the signal to the two other protein, and these two other proteins are the, so we have CDC6 and CDT1. Okay, now these two protein is activated, and we already explained that what are the abbreviation of these two protein. These two proteins is activated and they can bind here one and after other. Okay, so again we have, you know, the so let me draw here. So this is the first protein and we call that CDT1. So remember, we have two CDT1 can bind here. CDT1. Okay, and only one we have the CDC6, they can bind here. So let's say this is what, this is the CDC6. Now CDC6 can bind here. So after that, this CDT1, what are the function of this CDT1? They can, you know, induce the topological stress which is present in the DNA. So they can induce the topological stress. And CDC6 can activate the Mcm protein. Okay, Mcm, they can derive the Mcm protein. So that's why these two proteins are needed. So in the next stage, we have what? We have the Mcm protein is activated. So now at that specific region, the DNA is uncoiled, as you can see. At that specific region, the DNA is coiled. But when these two proteins are bind, so the DNA is now uncoiled. So after that, we have again the ORC, and we have 2 CDT1. This is also CDT1. And after that, we have what? We have the CDC6. Okay, with this CDC6, we have another protein activated. We call that Mcm protein. So then Mcm protein is also activated. So let's say this is what? This is the Mcm protein. So remember, we have 2 Mcm protein is activated at a time. Mcm. Okay. Now 2 Mcm protein is activated and they can bind on the opposite side of the ORC, origin of replication. They can bind at that specific, at the opposite side. Okay. Now once it can bind here, so then we get a complex. Protein complexes we get, and this complex is called as a pre-initiator complex. Pre-initiator complex. And this pre-initiator complex is usually in inactive form. So this is in inactive. And all this process is going on in G1 phase. So all these processes going on in G1 phase. G1 phase of the cell cycle. Okay. So remember, this ORC when bind, so it can bind at early G1 phase. This is very important. And when the complex is formed, which we call a pre-replication complex or pre-initiator complex, which is inactive form, it can form in late G1 phase of the cell cycle. Now, once we get this complex, then this G1 phase is replaced by the S1 phase. But remember, it is not directly replaced. So remember, in this pre-initiator complex, we have the DNA activator protein as well. And the DNA activator protein are what? The MCM and the CDT1. These two are what? These two are the DNA activator complex. activator complex. Okay, now they can just activate the DNA because the DNA are uncoiled, so it is possible by this complex. After that, once we get the pre-initiator complex, which is the inactive complex, then this complex need to be activated, and we have two proteins which is also activated, and these two proteins are called as CDK. Means these two protein are actually kinase protein, and these two protein are kinase protein, and we called it CDK and DDK protein. So two proteins are activated and DDK. Okay, this CDK can phosphorylate CDC6, while this DDK can phosphorylate the MCM. Now what does phosphorylation means? Phosphorylation means to activate, to regulate. Phosphorylation is very important in the cell cycle, cell growth, or in the cell life. Okay, so phosphorylation is very important. Why? Because phosphorylate can regulate all the activity of the cell. So once these two proteins are activated, so then the cell is going into S phase. Once it is regulated, then after that what actually happen? This CDT1 is also released out. So after that, the CDT1 is released out. Moreover, this ORC, and we have this CDC6, ORC and CDC6 is also released out, and it is also degraded. So these three proteins is released out, and these are the degraded. So after that we have the structures like that. So again, this is a DNA molecule. Okay, and we have just MCM. So let me draw MCM in a red color. So one MCM is present at that specific site, and one MCM is present. Okay, so MCM protein is present. And these are the hydrogen bond. Remember, okay, hydrogen bond is also present here. Okay, now what actually happened after that? The 2 MCM protein, we have just only 2 MCM protein is present at that specific site, and the rest of the protein is released out. Then after that, the ORC is also present at that specific site, but it is not in an active form. That's why I didn't draw here. Now, after that, some other protein is also activated. Now, this is the S1, this is the S phase of the cell cycle which is going on. Okay, so this is what this is the S phase is, S phase of the cell cycle. So in S phase of the cell cycle, we have the 2 MCM protein and ORC, which is in an activated form. So after that, we have some other proteins which are activated or recruited. These are the CDC45 and genes. But remember, this CDC45 and genes are recruited by the different kinds of protein, and these protein are the LD3 and SLD7 complex. So remember, these two complex can activate these two proteins, CDC45 and genes proteins. Now after that, we get another complex. So let me draw again the DNA molecule. So again, this is a DNA molecule, and in this DNA molecule we have again, this is the okay, so these are the MCM, and with this MCM we have the, this is the CDC45, and this green is genes. Okay, so it can make a complex. Let me draw here also. So we have also the C45 and the gens also. So these two proteins can bind here and it can make a complex. Now this complex is called as, now this is called as mature complex or activated complex. Now why it is called mature or activated complex? Because in this case we have what? We have the assembly of CMG. So we have the assembly of CMG. So again C stand for CDC45, M stand for MCM and G stand for gens. So this complex is called as activated complex. Now once the activated complex are formed, so then the DNA strand is melted, or this MCM is activated and they can act as a helicase, just like in prokaryotes, means DNA B protein just like acting as a helicase. So similarly in eukaryotes, the MCM is acting as a helicase and they can break the hydrogen bond between the two strands. So when the hydrogen bond is breaking between the two strands, so we get the replication fork as well as we get mean the two strands are completely separated from each other and we get a large replication bubble. So we get the the structure just like that. Now these are the replication fork and this is the replication bubble. So this is the replication bubble and this is the replication fork. So these two means complex, activated complex, or these two CMG can move in opposite direction. So one complex move in this direction while the other complex move in this direction, due to which the DNA is melted and we can get a replication fork, two replication fork and one replication bubble, just like that, as you can see in a diagram. Now again let me draw here in a small, this is what? This is the Mcm protein and this is another. Okay, so both Mcm protein is going in opposite direction. Now after that, once we get this means replication bubble, so after that other protein is also activated and these protein are the MCM 10 and CTF4. So these two proteins are activated and means these two proteins are recruited with this complex, and after that these two proteins can activate some other enzymes and we have polymerase, DNA polymerase, primase, and we have pCNA RPA. So these proteins are then activated. So first of all, primase is activated and they can add a few stretch of nucleotide, means they can add RNA primers. So here the RNA primers is added, as you can see. So it can be possible by the primase, and it can be possible with DNA G protein in the prokaryotes. Okay, so they can add the primer, and leading strand they can add just one primer. So the upper strand is the leading strand, leading, and the lower is the lagging. Try to remove the spelling mistake by yourself. Now at the lagging strand, the DNA primase can add many of the primers, as you can see. Why? Because one of the strand is going into 5' to 3' direction, while the other strand will be synthesized in 3 to 5' direction. Okay, so after that the polymerase alpha is activated. When the polymerase alpha is activated, so they can just add a few nucleotide, DNA nucleotide. So remember, this is very important. So they can just add a few DNA nucleotide. This red can show a few addition of DNA nucleotide and it is possible by the DNA polymerase alpha. Why? Because DNA polymerase delta cannot start the elongation. They can start the elongation, but they cannot start the elongation forcefully, or they cannot start the elongation whenever they don't detect this few stretch of the DNA nucleotide. Okay, and this few stretch of the DNA nucleotide will be possible by DNA polymerase alpha, and this DNA polymerase alpha will be activated when the RNA primer is present, and RNA primer will be aided by the primase enzyme. So this is very important. You guys need to focus here. In the prokaryotes, we just talked that we need only RNA primer, and then DNA polymerase can start the elongation process. But in this case, we need RNA primer as well as a few stretch of the DNA nucleotide as well. Now, once this process is done, then what actually happen? pCNA. So pCNA structure will be just like in this form. So pCNA is activated and they can bind here. Okay, it is a DNA sliding clamp, means they can DNA polymerase sliding clamp, DNA polymerase delta sliding clamp. They can slide the DNA polymerase delta to elongate the RNA primer, or to start the elongation process, just like that. Okay, now pCNA is also bound here, as well as it usually bound at the 3' hydroxyl end. Okay, 3' hydroxyl end. So it can bind at that specific point. Now after that, DNA polymerase delta is activated and they can bind to both of the strand. So they can hold both of the strand and start the elongation process. But remember this, there is, I forget one of the things. Before these primers attaching and stretch of the DNA nucleotide attaching, remember these two strands have a chances to self-anneal with each other. But this process can be stopped or prevented by another protein we call that RPA protein. So RPA protein can bind here with the DNA strand, with a single DNA strand, and they can prevent the self-annealing. When the self-annealing is happen again, so the DNA replication will be not going on. So RPA protein can activate it and they can stop this phenomena. And just like in prokaryotes, we talk SSB protein. Okay. After that, the whole process is going on as I explained. So then DNA polymerase delta is activated and they can bind with this pCNA, mean with this sliding clamp, and then they can hold the two strand and start the elongation process. How? We'll be talking inshallah in our next video lecture. So I hope these videos will help you, and I hope you understand about the initiation of DNA replication. But if still you have any kind of question related with this topic, you can write your question into a comment. Inshallah, I will give you answer as soon as possible. So like this video, share this video, and also subscribe the channel, and stay tuned for the elongation of DNA replication in eukaryotic. Thank you so much for watching.